our project to you. I'm Lilly Kuhn. I'm from Yarmouth, Maine.
I'm Clara McGurn. I'm from Lincolnville, Maine.
And I'm Ashley Gutierrez. I'm from Los Angeles, California, and this is our presentation on phytoplankton, pHun, fun.
All right. Phytoplankton are plankton, which are organisms that can swim up and down in the water column but cannot swim against the current. They photosynthesize through nutrition, and they are responsible for 50% of the oxygen in our atmosphere. They also have fixed 60% of the carbon dioxide in our atmosphere since the Industrial Revolution.
Ocean acidification. So ocean acidification is something that is barely beginning to get studied, and it's still poorly understood. But ocean acidification is the decrease in the pH of the ocean water. It's caused effectively by the absorbed CO2 that goes into the water. And so when the carbon dioxide is in the water, it creates a lot of chemical reactions that increase the hydrogen ions, and the increase in hydrogen ions then leads to the decrease in carbonate ions. And carbonate ions are crucial for the growth of the shellfish and the coral skeletons. So if the carbon ions are less abundant in the ocean, then that means that the shellfish and corals won't have a good time building their shells. And so humans there's a lot of factors for carbon dioxide in the atmosphere, and most of those factors come from humans ourselves because of the factories as shown in the picture, or the smog, or the change in land that is used, like deforestation. That's a factor of the change in land because a lot of the woods and the forests burn down. Also, like I said, smog in factories and the burning of fossil fuels. And these are factors of carbon dioxide and ocean acidification because they release carbon dioxide into the atmosphere and then it ends up in the ocean water.
Knowing what we did about phytoplankton and ocean acidification, we generated 2 research questions. The first being, what is the variation in pH in local coastal waters? And the second being, how does phytoplankton photosynthesis impact pH? Our hypothesis was that phytoplankton with access to light would photosynthesize and in turn increase the pH of the water.
To find out some more information about our question, we did some background research. And so dissolved oxygen, which was one of the factors that we're measuring in our question, is the amount of oxygen in water, or dissolved in water, relatively self-explanatory. And it's measured in milligrams per liter. And there's more oxygen in colder water than in warmer water. pH is a measure of acidity in water, and it's measured on a scale of 0 to 14, with 7 being neutral, where it's neither basic nor acidic, and above 7 is basic, and below 7 is acidic. So dissolved oxygen shows that there was photosynthesis happening in water because organisms that photosynthesize produce oxygen, and pH shows that respiration is happening in the water.
We plan to separate the samples into light and dark jars, the light being able to access light and the dark not being able to access light. And we plan to separate them because we wanted to see the difference or the change that the photosynthetic process has on the water, on the pH of the water.
All right, we collected samples from 3 different sites. We collected from Town Dock, Harris Secret River, and Little River. The Town Dock sample was saltwater and the other 2 were freshwater.
So the first step of our collections methods was we went out to all 3 sites on different days, and the days that we went out to them, there were different temperatures. Like when we went out to the Harris Secret, it was semi-cloudy. When we went out to the Town Dock, it was also cloudy. And when I went out to the Little River, it was the sun was out, but it was really cloudy. And so we used a 20-micron plankton net to collect a concentrated sample of water. And what we did was we set up a transect. Can't really see it, but it would be right here. The transect measuring 10 meters, and we drag the plankton net one time going this way and the other time going back, so that's a total of 20 meters. And then we put the concentrated sample in a gelato jar to take back to the lab. And we also took a 1 liter algae bottle to collect a normal sample of seawater and we used the concentrated sample to help us estimate the amount of phytoplankton cells in the algae bottle.
To calculate the amount of phytoplankton in our samples, we first calculated the volume of water that passed through our net as we were dragging it along the shore. And then we counted the number of phytoplankton in 5 different subsamples, and then found the average of that. And then we divided the number of phytoplankton by the volume of water to find the amount of phytoplankton in our 50 milliliter samples, which were the gelato jars.
And then to measure pH, salinity, dissolved oxygen, and temperature, we used probes that were plugged into a vernier. The photo on the left is of the probes, and they're in a freshwater sample. And we plugged them in, and then after taking the measurements, we split up the samples into 2 different jars. One had access to light, and one didn't. And we let them sit for 24 hours and retook the measurements at the end.
The results from our dissolved oxygen tests show that there was photosynthesis happening, which was really awesome because that's what we expected. On the y-axis is the location, and on the x-axis is the amount of dissolved oxygen in milligrams per liter. And the blue bars show the amount of dissolved oxygen after 24 hours in our light jars, and the red bars show it in our dark jars. And so it's clear that after the time that we set it to photosynthesize, there was more in the light jars than the dark jars, which did prove our hypothesis.
This graph shows the variations in the pH between sites, with the y-axis being the location and the x-axis being the pH. As you can see, the blue bar shows the initial measurement, which was before the 24 hours. The red bar shows the measurement after 24 hours in the light jar, and the orange bar shows the measurement after 24 hours in the dark jar. For the town dock, there wasn't much of a change in the pH levels before and after, so it was kind of an exception. Likewise, the Harris Seacon River showed very little difference in the pH levels. However, the Little River showed a big difference from the before and after for the pH levels. And that suggested that in the dark jar, since it decreased a lot from the initial measurement, that suggested that there was a lot of respiration occurring in the jar, and not only photosynthesis, which if there's a lot of respiration happening, that leads to the water becoming more acidic. And a trend that I observed when we were analyzing our results was that after 24 hours, almost all the jars were more acidic than they were just more acidic, and that suggested that there was a lot of respiration happening, even in the light jars, and not a lot of photosynthesis. Or it also suggested that micropredators were eating up the phytoplankton, which then leads to no photosynthesis in the jars.
Okay, this graph is showing the correlation between the pH and the dissolved oxygen of all of our samples, regardless of whether they were light samples, dark samples, or wherever they came from. A weakly fitted trend line that supports photosynthesis, because if the dissolved oxygen is increasing, it means that they are photosynthesizing and indeed spitting out dissolved oxygen. But we didn't really take into account respiration before we did the project. So the samples in the dark jars may have been respirating and spitting out CO2. So that might be why it's not as strong of a correlation.
Going back to our hypothesis, which was that the dissolved oxygen would be higher in the light jars, and that the pH would be less acidic in the light jars. The results from our dissolved oxygen tests show that there was photosynthesis happening, and that there was more of it happening in the light jars than in the dark jars, so that did support our hypothesis. But the pH levels in almost all the samples, and especially in the light samples, showed that it became more acidic after 24 hours, which wasn't what we expected.
Like any other project in the science community, there's always room for improvement in the project. And so one of our improvements that we could have made was controlling more variables, such as the temperature in the lab in which we did the measurement. And well, the temperature would be an important variable to control because we weren't aware that we were aware that the temperatures at the lab and the ocean water weren't the same, but we didn't take that into consideration. And so we thought that the phytoplankton weren't photosynthesizing as much because maybe they had to adjust to the temperature in the lab before they start photosynthesizing, because they're just really different temperatures. And collect more data, as in collect more data from each site, like have more samples from each site, so that we could compare our data more and make up like an average of like Freeport, or just have more data.
All right, we also wanted to have more time to leave our phytoplankton samples to sit so that they could photosynthesize and produce more dissolved oxygen. And we think that this would fully support our hypothesis if they have more time to respirate and photosynthesize. And we think that awareness of respiration would have helped us understand our data more thoroughly.
Further investigations, as we were doing the project, our curiosity expanded, and we wanted to learn more about it. And the question that I had, because we didn't observe the type of species that the phytoplankton were when we were observing them under the microscope, I thought that maybe the different species of phytoplankton affect the rate of photosynthesis. So I asked the question, how and like how fast do the different types of species affect the rate of photosynthesis?
I was wondering what other factors affected the pH of water, because we noticed that there wasn't a direct correlation between the phytoplankton number and the pH of water.
I was really interested in why the general level of pH in the local Maine coastal waters was so much more acidic than the global ocean average. And I was just wondering what effects might cause that. This is our citation slide and sources that we used in our presentation. And we want to thank Liz, Ann, and Kate, our amazing coastal adventure teachers, for being so supportive and enthusiastic throughout this whole project. And to our Res Life staff, Mary, Kate, Colleen, and Clara, and Zoe, for just being so awesome and always there to help us. And to all of you for being here tonight.
And taking an interest in our project. We're open to questions.
Yeah.
Was there a reason why you chose those 3 specific places, or was it like more of a random selecting process?
There wasn't a specific reason, but we just wanted a more general idea of what types of waters were.
And we were just those were the main sites that were available for us.
And also the town dock was obviously saltwater, while the 2 rivers were either partially saltwater and partially fresh, or just all fresh water. So that combination was interesting to us as well. Yeah.
Is there any reason to believe that there would be less respiration happening in the samples from the Harrisseekit River and the town dock?
For the Harrisseekit River, a lot of the results from that place were skewed, and our theory is that they may have all died, which is very unfortunate, but we think that might have been part of why that happened. Anyone else?